Welcome, students. This is Module 8, and Module 8 is going to be our last module on the carbohydrate pathways. And this one, we're going to be talking about gluconeogenesis, which is the creation of glucose. And so let me just go ahead and share my screen for the slides, and we'll get started here. Okay. That big. Okay, so the body has multiple backup redundant pathways because we have to have a continuous source of glucose for fuel, repair, and 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 synthesis of lots of things, like red blood cells. It starts with glucose. And so there's going to be periods where we're fasting, because we have dinner and then we don't eat again until breakfast, and maybe 12 or 14 or sometimes even 18 hours. So how do we ensure that all the organs and all the processes in the body get a continuous supply of glucose? Well, we have a backup plan that the body makes glucose, and it's made from non-carbohydrate sources. So, the other carbohydrate pathways, we're breaking down glucose and, we make glycogen and all this. But, this is one way that the body prepares for fasting. And, before it's going to be making glucose, it's going to deplete the glycogen stores first. And you probably will learn in other classes that usually within 72 hours of fasting or cutting out carbohydrates, the glycogen is pretty much depleted. Okay. And then glucose is made from precursors that are not carbohydrates, such as lactate, pyruvate, and glycerol, which is the backbone of triglycerides. And we'll get into that in probably, I think, modules 11 and 12. And also the alpha keto acids from the gluconeogenic amino acids. And the alpha keto acids is probably going to make more sense to you when you get into your 560D clinical biochemistry. But just know that gluconeogenesis uses non-carbohydrate sources, and it synthesizes glucose from them. And the major place that this happens is in the liver, but the kidney can also produce gluconeogenesis. And yeah. So you would think that, well, maybe we could just reverse glycolysis, but that doesn't really happen because the overall equilibrium of glycolysis really favors pyruvate formation, and it's really energy unfavorable to reverse glycolysis. Remember in the earlier lectures, we talked about the Gibbs free energy. Now, next module 9, we're going to be talking about the energy of the Krebs cycle and what regulates it. So the Krebs cycle has a very high negative G at the entrance. So that's very favorable to be pulling the substrates in, to be pulling the pyruvate to go to acetyl-CoA to go into the Krebs cycle. So it's not really going to go backwards. Gluconeogenesis requires both mitochondria and cytosolic enzymes, and we're going to go through that diagram step by step of how we start off making glucose. But I want you to know that during an overnight fast, most of this is occurring in the liver, and about 10% occurs in the kidney. But when we have prolonged fasting, it's really interesting because then now the kidney's really stepping up and it can become the major glucose producing organs, and it can contribute to maybe even 40% of the total glucose production. Okay, so I thought it would be easy to kind of understand the first few steps in gluconeogenesis by looking at this diagram that I got from another biochemistry book, Lippincott's. So let's go over here and look at step one. So this funky looking molecule, this blue part here is the pyruvate carboxylase. That's an enzyme, okay, that converts pyruvate into acetyl-CoA. So we have this enzyme here, and it's covalently attached to lysine. Now you'll find, like when you get in your later biochem courses, that lysine is, commonly at the end of a lot of the enzymes that attach a cofactor or a coenzyme or something to it. So that lysine is basically attached to biotin. So that's what you see here, step one. So we've got some biotin, we've got the attached to lysine, which is then attached to that enzyme. Now, going into this reaction, we have acetyl-CoA, and remember acetyl-CoA is attached to that B5. And so the acetyl-CoA is used here. And then we have carbon dioxide coming in, and it requires 1 ATP. Okay, so in this process, carbon dioxide, which you see here, is activated and it's transferred by this enzyme to the biotin's prosthetic group. So here you see, here's the CO2 here, and now it's attached to biotin. Now, the enzyme then transfers it to pyruvate. So here's pyruvate, and what we're going to do is put the CO2 onto pyruvate. Now pyruvate is changed into oxaloacetate. Now oxaloacetate. Now this is all happening inside the mitochondria. Right here, you see this. This diagram showed. This is the mitochondria, and then the gray part on the bottom part is the cytosol. Now we have to get this out of the mitochondria, but oxaloacetate, a lot of these, a lot of things inside the mitochondria are restricted. They're not allowed to flow freely in and out. So, and oxaloacetate is one of those things that cannot freely come out of the mitochondria. It cannot go in and it cannot come out. But NAD, which is, basically vitamin B3, it's the reduced form, which we have NAD plus an extra H, and we'll go into that a little bit more in Module 9. It reduces the oxaloacetate and turns it into malate. Now, malate can freely go in and out, and you'll be hearing about the malate shuttle in future lectures and also in future courses about the malate shuttle. So what this does is it regenerates NAD, and we'll learn in Module 9 that NAD plus is what fuels the Krebs cycle. So part of this process of creating glucose also regenerates NAD from the reduced NADH to the NAD that's oxidized form, and then that fuels the Krebs cycle, and that NAD is helping to make energy inside. So then now we have malate. Malate then comes out, and guess what? It gets oxidized, and it's turned back into oxaloacetate. Okay. Actually, it takes off 2 hydrogens off of malate and turns it back into oxaloacetate. Now, what happens is we have a high energy molecule, GTP, which is very similar to ATP, and carbon dioxide comes off, and it turns into phosphoenolpyruvate. And that's the very beginning steps of gluconeogenesis. And the from phosphoenolpyruvate, the rest of it is just the reverse of glycolysis. But this is a highly regulated process that it starts off in the mitochondria and then ends up in the cytosol. Now, the key steps that I want you to remember is, remember step one, biotin is covalently attached to the carboxyl carrier with lysine, okay? And it transports CO2 from the biotin carboxylase active site to the pyruvate carboxylate active site of an adjacent subunit using 1 ATP. So basically, we're putting CO2 onto pyruvate. So the biotin has that CO2 on its prosthetic group, and we're just taking it and putting it on pyruvate. Now pyruvate is turned into oxaloacetate. Now, since oxaloacetate can't cross the mitochondrial membrane, it's reduced to malate by NADH. So then malate can then cross over, and now we're in the cytosol. Okay, and then remember, we're regenerating that NAD that helps run the Krebs cycle. And then in the cytosol, malate is oxidized back into oxaloacetate, which is then oxidatively decarboxylated, giving off CO2 and using one high energy GTP, and it's turned into PEP. So remember these key points. Oxaloacetate can also be converted to aspartate, which can leave the mitochondria. PEP is the critical precursor for gluconeogenesis. And oxaloacetate cannot cross the mitochondria. It can't go in and it can't come out. And this pathway gives us a backup way to produce PEP. And the rest of the pathway is simply a reverse of glycolysis. Now, this is a pretty scary looking pathway, but I just put it here because I really want you to focus on the energy requirement of gluconeogenesis. Look at it, it requires 4 ATP, 2 GTP, and 2 NADH. But if glycerol enters the gluconeogenic pathway, then there really isn't any glucose, no energy requirement. So here, we have lactate, and some amino acids going in to make some pyruvate and also, and then it's converted to oxaloacetate. Then we have this PEP, and then the rest we already went through in the previous lectures on glycolysis, because from here it's basically a reverse. Now, I want you to look at this diagram. On the left, I guess it's kind of a peach color, pink, and on the right is blue. So glycolysis, which we already studied, is exergonic, right? We're breaking down glucose and we're making energy. And then and that happens mainly in the muscle and the brain. And then gluconeogenesis happens mainly in the liver. So we're making glucose. So on the one hand, we're breaking down glucose, and on the other hand, we're building up glucose, okay? So in the gluconeogenic pathway, let's look real closely here. This fructose 6-phosphate is made by the enzymatic reaction of fructose 1,6-phosphate here, okay? And that fructose 6-phosphate is easily converted into glucose 6-phosphate. So in gluconeogenesis, we don't make glucose directly, and there's a reason for that. Glucose 6-phosphate is pretty much the end product of gluconeogenesis. And you might wonder, well, why aren't we just making glucose? Well, since gluconeogenesis occurs in the liver, then the liver is the metabolic hub, okay? It's going to control glucose throughout the whole body, okay? And so the liver has has an enzyme, there's an enzyme called glucose 6 phosphatase. And you see right here at the very top of this blue one, I know it's kind of small writing, but if you enlarged it, this glucose 6 phosphatase turns that glucose 6 phosphate into glucose, and that is it's very easy, and it catalyzes it is what allows us to make glucose. But why is that enzyme only in the liver, mainly in the liver? It doesn't have the muscle and doesn't have that enzyme. So the phosphatase is only present in tissues whose job is to maintain energy homeostasis in the body. So, which basically is the liver and the kidneys to a lesser extent. Okay, and this allows glucose to go anywhere. So this allows preferential handling of glucose so it can be supplied to the brain as well as to the muscle and the red blood cells. And by contradistinction, the muscle doesn't contain this enzyme, so it can't be converted to the glucose 6 phosphate can't be converted to glucose for use in the muscle. And you think about it, and it just kind of makes sense, because what if you're like running down the street or you're doing some high, aerobic activity, and you've depleted your glycogen stores, and you're running on fuels, and so then you have this glucose 6 phosphatase, this glucose 6 phosphate in the muscle. If you had that enzyme in the muscle, that muscle would be burning up your G6P into glucose so you could keep playing. But what's going to happen to the brain? All of a sudden, your brain is devoid of energy because all this muscle is taking up all this glucose for energy, and so you might just fall down dead. So this is this enzyme allows the liver to be called the metabolic hub. Okay. And so now in the muscle, the glucose 6P can be converted to glucose 1P for storage as glycogen, and eventually retrieved as an energy source for the muscle use only. So in the muscle, it's converted to G1P, but in the liver, we have that glucose 6 phosphatase that allows it to be converted to glucose, providing energy to all kind of pathways that we need. Okay. And so these two things, glycolysis and glucogenesis, they're happening all the time, and they're going back and forth and back and forth. So we need to talk about, well, what's regulating? How is the body going to decide whether we're going to do glycolysis or gluconeogenesis? And that's decided by the energy of the cell. Okay. So when the energy of the cell is low, think about it, there's very little glucose, then glycolysis is going to prevail, providing that we have enough, carbohydrates, right? So glycolysis is going to prevail. What if we just had a big plate of pasta and we're just kind of sitting around watching TV? The energy of the cell is high. So what's going to happen is the gluconeogenesis, because, we've got all of this excess storage. Glycolysis, when the energy of the cell is high, but the gluconeogenesis is going to take lactate, amino acids, and those, pyruvate, and it's going to put it into gluconeogenesis and possibly use it as storage for fuel for later. Okay, so when the energy of the cell is low, glycolysis prevails. When the energy of the cell is high, gluconeogenesis prevails. Now, let's look at this pyruvate and lactate. That's right at the entrance of the mitochondria, of the Krebs cycle. So I put the two diagrams on the left is pyruvate and the right is lactate. And pyruvate and lactate can go back and forth, back and forth, and back and forth. And we'll talk about, what regulates that in a minute. But pyruvate gets converted to lactate when we have a lot of acid around. And remember, hydrogen ions are acid. So if we got a lot of hydrogen ions around, that NAD is going to exist mostly in the reduced state. So NAD will donate a hydrogen ion to pyruvate and convert it to lactate. And then lactate is a precursor for gluconeogenesis. So where does the body get the lactate from? High intensity exercise, anaerobic metabolism, cells that lack mitochondria, such as our red blood cells, produce lactate, and also in the Cori cycle. And I put the diagram here of the Cori cycle where glucose goes into the muscle, gets used, it's turned into lactate. And when you're exercising and you get that burn, that's that lactate buildup. So the lactate is taken back to the liver and turned into glucose. So then that's called the Cori cycle, where we actually can use lactate as a fuel. Okay. Another important conversion is the source of lactate comes from the conversion of pyruvate to lactate. And you'll note that with lactate, the OH group on the number 2 carbon has an OH group in its reduced form of pyruvate. So here's the OH. See how you have two OH groups here. So one of these double bond O's is reduced when the hydrogen is added. Okay, and okay, let's go to the next slide. So I added this slide here. This is from my course that I teach in organic acids, that lactate and pyruvate are the principal products of carbohydrate metabolism, okay? And they can go back and forth, back and forth. And so here you have somebody eating carbohydrates, and then also we have glucose and other sugars. They go to make pyruvate and lactate, and they can go back and forth, and it's reversible. And then it's acted on by the pyruvate dehydrogenase complex to make acetyl-CoA, and then that goes into the Krebs cycle. And then on the left here, we have pyruvate. This is the oxidized form, and then lactate is the reduced form. Now, there's several things I want you to remember about pyruvate and lactate is that we make gluconeogenesis from lactate, okay? And these are anaerobic products of glucose breakdown from glycolysis. This is all in the cytosol, okay? And then under anaerobic conditions, pyruvate will convert to lactate by an enzyme called lactate dehydrogenase. And that will reproduce NAD needed for the citric acid cycle. Now, in your future practice, you might see lactate dehydrogenase enzyme. And so I want you to know that what they're measuring here is how much lactate is being produced, because in certain metabolic states, especially like cancer, where the mitochondria gets kind of shut down by the cancer cells, and then pyruvate gets shifted to lactate, well, you'll see an increase of the LDH enzyme, lactate dehydrogenase, and that. So basically, when you have a lot of lactate or a lot of lactic acid, your body's very, very acidic, and everything is sitting in the reduced form. And so, and all this oxidation, not reduction that's going on in the body comes to a standstill because we have too much acid, too many hydrogen ions sitting on that receptor, and they're unable to keep it going and pass it. Okay. So oxygen is is a very important factor on whether pyruvate's going to convert to lactate or not too. So when we have a lot of oxygen aerobic conditions, the glucose is going to convert to pyruvate. Okay, it's going to be, and then pyruvate will quickly be converted to acetyl-CoA, and it's going to avoid lactate buildup. Okay. And so pyruvate is always going to prevail when we have a lot of oxygen availability for the mitochondria. So when in anaerobic conditions, lactate will always prevail, okay? Because the mitochondria is an aerobic environment and is oxygen requiring, okay? And then we're using the enzyme that we used to convert pyruvate to acetyl-CoA is called the pyruvate dehydrogenase enzyme complex, which you'll learn later. Now, this isn't something you're going to be tested on, but you will learn this in 560D about the difference between D-lactate and L-lactate. If you remember from organic chemistry, D means dextrorotary. That means the rotation of the molecule around the central heavy point of the molecule. It's gonna spin to the right. And L-lactate, it spins to the left. Okay. Now, L-lactate is produced by human metabolism. That's the lactate that our body produces. Now, the bacteria living in our gut produce D-lactate. And so when you get later on in your career or later on in the program, you'll learn the difference between D-lactate and L-lactate. And the D-lactate can be absorbed from the gastrointestinal tract and combined with L-lactate and make your lactic acid levels go much higher. And many of the functional labs will distinguish between D-lactate and L-lactate. But I do want you to remember that when we're making lactate, one of the principal reasons is to regenerate the NAD plus that helps to fuel the Krebs cycle. Okay. Notice that the enzyme lactate dehydrogenase is using that cofactor NAD. Now that's the most important regulator of pyruvate and lactate. So in conditions when the pH of the blood is low, then there's a lot of hydrogen ions. And remember I said that in the last slide, when you're really acidic, they're going to be sitting on that NAD. Well, there's not a whole lot of NAD in the body. And when we're acidic, all those hydrogen ions are going to be attached to that NAD. And then now it's not going to be able to pass it anywhere. Okay. And so the pyruvate is going to automatically convert to lactate in order to help regenerate that NAD for the mitochondria. And then the lactate then now can go through gluconeogenesis, and the NAD will be freed up for that Krebs cycle. And we'll talk more about the Krebs cycle next week. So in other words, the ratio of the NADH and the NAD is, if it's low, then pyruvate will prevail. Okay, if the ratio is high, then lactate will prevail because lactate dehydrogenase is bidirectional. Okay, now the first step is to take pyruvate from whatever source, either lactate or glucose, and transport it into the mitochondria. And in the bottom of the diagram on the right, you can see that pyruvate interconverts to lactate. You can see the bidirectional arrows over here. Okay. And the reason why we don't just reverse pyruvate into PEP, which is, at the last step in glycolysis, is because the pyruvate kinase of glycolysis is not reversible. And no enzyme exists in the cytosol for the reversal process. Okay. But in the mitochondrial matrix, that enzyme pyruvate carboxylase that adds that CO2 to the 3-carbon pyruvate to form that 4-carbon oxaloacetate. Remember, and it costs us an ATP. That enzyme requires biotin as a coenzyme. So biotin is a really important part of energy regulation, and we'll get into that in a later lecture. Okay, so that is the end of part 1. And in part 2, we're going to talk more about energy homeostasis. And really, that's what the whole course is about, is, using fats, carbohydrates, and proteins to regulate the energy in the body. I mean, the body is built so perfectly that it's always trying to achieve homeostasis, and there's all these backup mechanisms and back doors and all of that. And when you get into your later courses, you'll learn how to measure that on test, and then this course gives you that backbone to in your head, to where some of these pathways will start making a whole lot of sense, to where you'll understand the connection of this course to all the rest of the courses, and then understanding any energy homeostasis. So a lot of diseases have to do, or even illnesses or symptoms from people, have to do with cell membrane damage, the things not getting in and out, nutrient deficiency, and the body's trying to compensate from one pathway to another, trying to provide a steady source of energy. And so we'll talk more about the regulation of all of that in part 2. See you later.